Craig, I was reflecting this morning and it occurred to me that it's now the 20th anniversary of the paper that Kim Huber and I published suggesting the outlandish possibility that there might be the potential for a small molecule therapy to change the course of Fragile X syndrome. It's been quite a ride the last 20 years. A lot of high hopes, and some of them have been dashed, but I feel like we're actually in a really good place to, accomplish our mission sooner rather than later.
Yeah, I mean, I really enjoyed joining that ride where I came from the autism field where, no single gene disorders, no clear leads, massive heterogeneity, and moving into Fragile X with the concept of being able to conduct animal studies, to do bench lab studies, and then truly translate those findings into human work. And we've learned a heck of a lot in the last 2 decades. And, in my mind, we think a lot about failed trials, but every quote failure is a lesson learned. And, overall, our lab and our human work is really focused now on Fragile X is one condition, but how do we define variation molecularly, electrophysiologically, functionally, and then match individuals with treatment?
Yeah, I completely agree with you. When we started, there's very little experience in running clinical trials, and certainly not to the standard that would be required by the FDA, and so many unknowns that occur with any drug development program, like what's the right target? What's the right compound? What's the right dose? Are there going to be dose-limiting side effects that we have to take into consideration? Who are the correct patients? What are the right endpoints to measure? But I completely agree with what you're saying, is that every time we do a trial, we learn something new, and we apply that knowledge. So it's very iterative. And I actually share your optimism that we have learned enough now to actually deliver in the near term.
So in our lab and other labs, high density electrophysiology or brain electrical activity, and what are small molecules doing early in phase 1 and phase 2 studies to see target engagement. And we've really moved away from a reliance on pen and paper measures. Now, we think that's huge and will allow us to truly link preclinical to clinical outcomes. But, even that has taken a lot of time. And again, hindsight is 20/20, and I don't think we don't view it as setbacks, but it's really a new generation and a new iteration of trial approaches that are more tightly linked to animal experiments.
Yeah, I hear you. I mean, I think that there's a lot of flying blind, to use another flight metaphor, but if you don't know, you don't have a good biomarker of target engagement, then it's a lot of guesswork to know, like, what's the right dose? Does the therapeutic effect persist, or is there a development of acquired treatment resistance, which it turns out to be a potential problem? And that knowledge would give you so much confidence to say, for example, in the case of your work, there's an electrophysiological signature of altered function. And this signature can be modified by a treatment, and you can learn what is the correct dose to move that biomarker. From the beginning, Fragile X could be viewed, at least some aspects, as a metabolic brain disease, to the extent that it's a dysregulated protein synthesis in neurons. And of course, we now understand that that's a process, a pathophysiological process that contributes to some symptoms. There are other pathophysiological processes like altered ion channel function, particularly potassium channel function, and still other features that are due to simply altered brain development. The wiring diagram is different. And so we would not expect one treatment to hit all of those 3 causes of dysfunction in Fragile X. And having some insight from a biomarker on, what symptoms line up best with what treatment strategies would be super helpful. Because, you could have a fantastic treatment, but you're measuring the wrong thing. And I think that has been some of the clinical experience, which is very frustrating to the clinicians and the families.
And not to mention layering development into that, where we think about treating, from newborns to toddlers to adolescents to adults, and, where your treatment windows there are. So, when we hear of positive leads in preclinical, data, and we take that into human studies, I think, we've definitely sharpened our tools in quantitative ways, but we also have to look at developmental windows and cohorts maybe being different. And the idea that if we have to study something in adults first and we do signal finding, then we're systematically moving through the ages. We're thinking about males and females differently, so they're both represented. We want to represent everyone, but if we don't control our studies to be able to power and think about subgroups with signal detection, I think we're doomed to fail.
Yeah, that is a really good point. Also, then I'm also amazed actually by the work that came out of Peter Kine's lab in Edinburgh, where they found that timely treatment during development could lead to an improvement that persisted for many, many months in the absence of continual treatment. Like it was intervening at the right moment in development could alter the course of brain development in a very effective way to help a long-term outcome. So, I really agree with you, Craig, that we have learned so much, and I feel that we really are on the threshold of being able to apply this knowledge to develop some medicines that are going to help.
I feel like we're finally at the honest point where we interface with treatment developers, around the world, where we can take a small molecule, combination of small molecules in our hands, use all the tools that we've developed to detect signal in a meaningful way. And what's really surprised me in the last 5 years through our work with single dose challenge studies, phase 1A designs with 10 to 24, patients, that it doesn't take huge samples to detect signal when you're using electrophysiology, when you're using computer-based testing, which I think is really great for anyone that thinks about drug development in this field, because your ability to make go, no-go decisions using quantitative measures and then de-risk your phase 2 and phase 3 programs once you have a sense of who the responders are quantitatively really just opens the field up in ways that we've not had before, where companies and other developers faced funding large phase 2 programs with heterogeneous samples, males and females, mosaics, non-mosaics. We're just not in that place anymore because of our tool development, because what we've learned and about how we've improved our linkages to preclinical studies. I mean, a lot of folks now can do preclinical FMR1 knockout mouse electrophysiologic studies, see what their compounds are doing in the mouse, then immediately move into a 1B target engagement study in humans, see if there's synchrony and see which humans are responding, even with small subgroups, because we don't have placebo response with those measures in different ways. So we're very excited about that. And, despite all the quote failures, we're more excited in our lab than I've ever been in my career, because these tools are really poised for success because of, as you said, Mark, everything we've learned from the past.
Yeah, it's been really gratifying to see that sort of these small new clinical candidates keep being introduced. And I think your point is also very well taken, which is that we, with a rare disease like Fragile X, we don't have the luxury of endless attempts to succeed, because clinical trials are so expensive that it's not like a huge indication like major depressive disorder or schizophrenia, where you keep your Alzheimer's disease, where you just keep throwing things at the wall in the hopes that something sticks. We don't have that luxury in Fragile X. So the more informed we can be going into doing the study, the more likely we are going to be to succeed. So thank you for advancing the field in that way, Craig.
Yeah, and I think it's important for us as, human and translational scientists, we use what we learned from, example, our NIH studies to inform our treatment development.
Another feature of Fragile X that always impressed me from the beginning was the evolutionary conservation of some of the pathophysiological mechanisms, literally from fruit flies through mice and rats and humans. And so there is I'm very bullish. I mean, perhaps because I'm a basic scientist at heart, but I'm a firm believer in the relevance of the animal models, particularly in a genetically defined disease such as Fragile X.
And we think about who with Fragile X is best modeled by the available preclinical model. So if it's a knockout mouse, who is best modeled? When we do human high density EEG and we look at an individual signal, is that a signal that matches the electrophysiologic pathophysiology we see in the mouse or not? Is that someone who is going to be best modeled? That's how we think about it now. There's still great utility, but obviously it's not an exact replica of the human experience across the board.
Yeah, I think that's cool. And then the question will become, what is the best behavioral correlate or maladaptive behavioral correlate of the electrophysiological signature? Because then that gets you closer to the quality of life objective. But I would say also on the human side of saying you have an altered EEG signature, what major behavioral domain does that correlate with? Is it irritability, aggression, and so on and so forth? And that's another, objective or challenge that we have.
Not to mention thinking about anxiety, which we all believe is a core lifelong feature, but very difficult to measure. And we're putting a lot of effort in collaboratively across human labs to think about anxiety more objectively and more consistently, because we get regulatory feedback of this is a developmental disorder. People can't self-report anxiety. So how do you measure it? But it's so important to the phenotype. So that's something we really have to sharpen tools in that area to have a chance to think of it as a target, because we all know clinically that if you significantly reduce anxiety, quality of life and function comes with it. But if we can't define it in a logical, consistent manner, how do we use it?
Yeah, I think we still have obstacles to clear to go from where we are today to getting a medicine approved. And of course, one of those is the continual need to educate the FDA and the regulatory authorities on the urgent need and the very fact that it won't be one size fits all.
In my last 15 years interfacing with the FDA, I think they're better positioned now to understand variation in Fragile X, understanding these quantitative measures we discussed. Not that they're going to be primary approvable outcomes, but they can have utility in defining subgroups and understanding at a regulatory level that, yes, you can have an indication for some individuals with a single gene disorder, and that's okay if you're defining them appropriately. So I think that backdrop is going to be key to enabling everything we've discussed.
Well, let's hope that we can close the deal in the near future.
I think we're getting close. This was super enjoyable, Mark. Really enjoyed the discussion.